Thanks, Jeremy, for the very kind introduction. And in the spirit of this conference, I called my talk Mastering Pharmacokinetics and Immunogenicity of Bispecific Antibodies. So what I want to do, I want to show you a case study, a novel anti-TNF IL-17 bispecific antibody construct. And I want to share with you some special considerations when it comes to pharmacokinetics and immunogenicity. And I want to do this by quickly introducing the technology, the lead compound, and then actually touching on what's our testing strategy. How do the assays look like? And then, of course, I want to show you some case data. So, and since we are a rather small group, whenever you have a question, please go for it. Don't wait until the end of the talk, just shoot for it. So, what are phenomers? What's the technology? So, phenomers are alternative binding proteins with antibody-like features, but there are no antibodies. They are derived from a protein tyrosine kinase called Fyn kinase, and I should better say they are derived from the so-called SH3 domain. If you zoom into this domain, this is simply enlargement of the SH3 domain. There are two flexible loops, the SARC loop and the RT loop. And those two loops, they are responsible for protein-protein interaction. They grab for other proteins, like the CDRs of monoclonal antibodies. So what we did, we simply did permutate all the amino acids in these flexible loops, generated a phage display library, and out of this library, you can fish binders against your target of interest. So that's the technology of phenomers. And so the characteristics of phenomers, they are fully human because they're derived from a fully human protein, and this so-called SH3 domain, it's fully conserved across mammalian species. So they are small and they are less complex. So that I like this, being small and simple. And there's one advantage that did not even show on here. This is you can easily fuse or co-express phenomers with other proteins. And this brings us to the next technology, to the so-called FynomAb technology. And that's quite easy. Whenever you fuse or co-express a phenomer to a monoclonal antibody, to a mAb, the resulting end product is we call a PhynomAb. And this is just some cartoons how such PhynomAbs look like. So you have the classical IgG backbone and then the blue little circles that represent the phenomers. And as a consequence, those molecules now are bispecific, or you can even generate tri- or multispecific molecules. So hopefully I can convince you later on that those molecules, they are stable in vivo and that they still retain their activity, so their binding characteristics. And we are quite flexible to which ends we can fuse our phenomers. So this is maybe not so important for our case study, which is a dual cytokine inhibitor, just neutralizing two soluble targets. But this may be very well important, for example, if you want to target cell surface molecules. Then the architecture, in other words, where do I fuse the phenomers, can play a big role. And the big advantage of going piggyback to an antibody is that we now have antibody-like pharmacokinetics. And that really helped. Okay, so now the PhynomAb technology brings us to our case study, to the molecule I want to show you. This molecule is bispecific for TNF and IL-17, and the backbone, the monoclonal antibody backbone, basically is adalimumab, or I should better say it's the amino acid sequence of adalimumab. And then there are two C-terminal light chain fusions of phenomers that bring in the specificity for IL-17. So for this talk, I don't want to go into the rationale, why did we combine TNF and IL-17. Hopefully you believe me, there's good reason to do this, and we have a lot of pharmacology data that actually shows this. I just have 3 slides to show you in essence, that the molecule does what it's supposed to do, and I start with binding to each of its targets separately. And we did include reference antibodies. We did include a reference antibody called secukinumab from Novartis, which is anti-IL-17 specific as a benchmark for binding to 17, and of course adalimumab as a benchmark for binding to TNF. So if you look at just the binding constants, the KD values, so we are in the picomolar range, and with respect to IL-17, this molecule is approximately 2 to 3-fold, does it bind stronger as compared to secukinumab, and with respect to TNF binding, we are in the same range as adalimumab, and you would expect this because adalimumab is our backbone. And the same is true in cellular bioactivity assays, that we are slightly more potent as compared to secukinumab, but there's the same potency as compared to adalimumab. So this is just showing you that if you look at each of its binding arms separately, that the molecule can bind and is bioactive. But I think what we have to show also is that the molecule can simultaneously bind. Yeah?
Invited interruptions?
Sure.
So you say you have a data molecule from the signaling events that is more specific than an antibody?
So this molecule in a way combines dual specificity. You could compare it like if you would mix adalimumab and secukinumab, 2 monoclonal antibodies. This is basically the mode of action of this molecule, but you have just one molecule. You just have, from a production, from all that stuff, just...
I'm surprised that your IL-17 specificity seems to be better.
So we have, that's actually not shown on this slide. It's just shown that the binding affinity and the potency in neutralizing IL-17A, if you do a head-to-head comparison, secukinumab and our molecule, we need less of the molecule. It's slightly more potent. I'm not touching on specificity, but since you are asking, it also looks like that we are a bit more specific for IL-17A. So there's IL-17A and F and heterodimers. So with potency, we are slightly more potent. If this is the answer.
It's very impressive.
Okay. Alrighty. So here we wanted to address the question: can we simultaneously bind? And this is just a biocore experiment where IL-17A is bound on the plate, and then you add the molecule. You can see you get an increase in mass. So it's binding to IL-17, and then when we add TNF on top, we do see another increase. So that tells you the molecule can simultaneously bind to both its targets. But I think what's even more important is that you can show that you get bioactivity to both targets simultaneously. And to show this, I want to introduce our bioactivity assay first. So that assay works that we use Ag29, that's human colon carcinoma cell line. If you stimulate Ag29 cells with TNF or IL-17, they release a chemokine, GRO-alpha. So that's your readout, release of GRO-alpha. If you now pre-incubate with COBA322 and you get neutralization of TNF, neutralization of IL-17, you should have no or just a low release of GRO-alpha. This is how the assay looks like. So let's start with the controls because that's a complex set. So if we stimulate the cells with just media, there's no release. Makes sense. If you stimulate with IL-17 only, we get some GRO-alpha release. If we use TNF, it's better. If you have both, then you get the maximal response. So that's your starting point, your maximal GRO-alpha release. And now we did a titration with secukinumab, and you do see you get a dose-dependent decrease in your signal. Not to 0, because your contribution of IL-17 is just this bit. Makes sense. If we go with adalimumab, we also get a dose-dependent decrease. Now it's stronger because the contribution of TNF is more. And if you now mix adalimumab and secukinumab, you get it down to 0. And the same is true with our compound. So that tells you two things. First of all, we can show that we can simultaneously neutralize TNF and IL-17, but it also shows you that with one molecule, we are as potent as a combination of two monoclonal antibodies. So that was just to show you that the molecule is doing what it is supposed to do. And now actually I switch gears to special considerations when you have such a molecule in terms of pharmacokinetics and immunogenicity. And this is just high level. I want to mention here, depending on how you set up your assay, you can just follow something that has a human FC part, or you can try to come up with sophisticated assays to show that the molecule is intact or whether it's bioactive. And we have generated such assays, and hopefully I can convince you that the assays do actually sense for intact and bioactive molecule. Also in terms of immunogenicity, in addition to the classical immunogenicity assay, we also came up with an assay where we can, in a way, show what is the specificity of anti-drug antibodies, or which domains are they binding to. So now I start with our testing strategy. So for all our early clinical study samples, we actually start with PK measurements with two different PK methods. And I'll show you the assays later on. And then we do the classical stuff. We do an ADA screening assay, confirmatory assay. Then we do titer analysis. But then we also do this so-called specificity analysis. And now I start with PK assays with our first assay, and that assay is called monofunctional assay. So why is it called monofunctional? So that assay uses TNF as a reagent. So it relies on binding to TNF, so binding to one functional unit of the molecule. So that's why it's called monofunctional. And by using TNF with a label and an anti-idiotypic antibody with a label, you get a complex formed and you can detect this complex. So this assay only works if the molecule still can bind to TNF-alpha. And the second assay is called bifunctional. And actually, it's quite similar how it looks like. The only thing is that now we also use IL-17 as a reagent. So in other words, this assay only works if both entities, the anti-TNF binding entity and the anti-IL-17 entity, is present and biologically active. So now the advantage of having two different assays is that when you do sample analysis with both that assays and you get the same signal, then that tells you indirectly that you have no degradation of your FynomAb part. There could be cleavage of the FynomAbs and then the IL-17 functionality is gone. If you do this with that PK stuff and you get the same signal, then you know, okay, it's still there. And I just show you now some data. This is data from our phase 1 study. It's a single intravenous infusion in psoriatic patients, and it's here 4 different dose levels. So the starting dose is 0.03 mg per kg, and the highest dose shown here is 1 mg per kg. And actually you see 4 different lines representing the 4 different dose levels. But here you can actually see 8 lines, because per dose group we did test with our 2 PK assays. That's good because the assay results, they are superimposable. And that tells you that actually there is no degradation in vivo. So what we can see here is fully functional molecule that is present in vivo. Okay, now let's move from PK to immunogenicity assay. And this looks a bit complex, but for those who deal with immunogenicity assays, this is just a classical bridging format. So we start with a complex of your anti-drug antibodies and CoVA322, or just anti-drug antibodies. Then we have an acid dissociation step in case we have still a complex here, that we get this apart. And then we work with CoVA322 with a biotin label and CoVA322 with a sulfur tag. That is forming a bridge with your anti-drug antibody. So that's why it's called a bridging format. And then you get your signal. And the screening assay is in a way set up that you get 5% of false positive. You want to be hypersensitive This is according to the guideline. And then you do in a second step, when you get positive results in your screening assay, a confirmatory assay, where you actually use CoVA 322 without a label, and that works as a competitor. And then you get your signal down, and this assay is designed in a way that you then get 99.9% positives. So on the next slide, I just show you the parameters that we looked at during validation, and that's too detailed. I just want to show you that with respect to drug tolerance or drug interference, that we did test this at 2 different concentrations of the positive control at the limit of detection at 10 nanograms per milliliter. We have a drug tolerance level of 10 micrograms. And if you go to 250, which is over the thumb, the threshold that the FDA claims for your sensitivity, we are even at 100 micrograms. And this is far above what we would expect to see in clinical studies as a trough level. So in other words, this assay is sensitive and drug tolerant enough for the indication or for the dosages we are using. And maybe I can, we do not see an interference of the target. And this may not sound intuitively when you look at an ADA assay and the influence of your target. So maybe I go one back. It's a bridging format. So the anti-drug antibody is forming a bridge between your molecule. If you now have a target like TNF, which is a trimer, or IL-17, which is a dimer, and you have a risk of carrying over your targets into your assay, it could well be that you have no ADAs, but you have your target, and the target can form a bridge because it has 2 or 3 units. So that's why you are better off of testing that this is not happening. And we did test this, and this assay was not sensitive to the presence of target. So the assay, in a way, is working quite well. And then we came up with an exploratory assay to look for IgM, which is just an easy ELISA where our molecule is on the plate. Then you have your IgM that is specific and you detect via anti-IgM. And I think a more interesting assay is now an assay to dissect the specificity. The concept again is bridging. So your ADA bridges 2 CoVA 322 molecules. The only difference is that we do have a pre-incubation with a competitor. And if this competitor is specific, it binds and blocks the ADA and you get a reduction in signal. And this assay is only as good as the reagents you have to use as a competitor. So of course we use CoVA 322 as a competitor because that has to decrease the signal, otherwise it wouldn't be specific. And if you now just virtually think, if you rip apart CoVA 322, you would get adalimumab as a backbone, and you would get the specific FynomAb on its own. And then we have an irrelevant FynomAb. Actually, that's the SH3 domain, so that's the scaffold on its own as a negative control. And we have a human IgG1 irrelevant antibody as a negative control. And then we have 2 so-called FynomAb controls. One is called FynomAb control adalimumab because it has adalimumab as a backbone, but now it has the wild-type FynomAb, not the specific one. But the domain arrangement and the linker is the same. And the last control is the other way around. It's an irrelevant human IgG backbone, but now the relevant FynomAb. Okay, so when we established this assay, we did not have human samples available, but we did have sample from monkeys available that were ADA positive during PK studies, and that we then did heavily immunize with CoVA 322 formulated with an adjuvant to generate a high titer positive control for the validation of our ADA assay. And we asked the question, okay, how does the specificity look in such a, let's say, worst case scenario for immunogenicity? And this is shown over here. So this is your signal without any competitor. So that's your starting point, and you can see the standard deviation range. So now if you use COVA322 as a competitor at 3 different concentrations, you get the signal down to 0. And this is, what you would expect. That's your positive control that the assay works. The same is true if you just add adalimumab plus the specific FynomAb, you also get it down to 0. And also with adalimumab on its own, the majority of your signal is blocked. And the same is true for this so-called FynomAb control that has adalimumab as a backbone. All the other controls, basically they may have some subtle effects, but that's not really different from your, let's say, standard deviation. So I know that we cannot use any animal data to predict anything to humans, but what that tells you, in a given species under harsh conditions, you get basically an immune response that's more directed against the backbone and not against the specific FynomAbs. And now I have 2 slides where I try to bring all the assays together with a human case example or with clinical data. And I start off with a subject that we call late ADA response. So I start with the colored lines, red and blue. This is again pharmacokinetics, mono and bifunctional assays. And just to remember, it's single intravenous infusion. So up to the first 6 weeks, it looks brilliant. It's very nice pharmacokinetics. This is what you would expect. Then between week 6 and 8, you do see accelerated clearance. And at this time, in the anti-drug antibody assay, it starts to be positive and the titer increases, although the titer on its own is actually quite low. But it matches, your drop in PK induction of anti-drug antibodies. So then we ask the question, okay, is there IgM involved? And you can see that for this particular one, there's no IgM signal. And then we did subject this sample to our so-called specificity assessment. Again, this is your starting point. That's your signal. Again, with COVA322, we get it down to 0. With adalimumab plus the FynomAb down to 0, adalimumab on its own is doing a very strong inhibition, and the same is true for adalimumab-based FynomAb control, and for all the other controls, there is no effect. So if I were to draw a black and white conclusion on the specificity for this patient, I would say this patient experienced an adalimumab response and not a FynomAb response. I'm also saying here that this is high affinity interaction. I will show you in the next slide why I think it's high affinity. It took us only low concentrations of competitor. There's no IgM, and we call it late because it took quite some time until we did see the anti-drug antibodies, and we knew that our drug tolerance level is high enough. So that's not an artifact of the assay. This is true that it only late kicks in. So now I show you the other extreme. This is now a subject where we did early on see an effect on PK and anti-drug antibodies. So again, for the first 10 days, it looks as expected. But by week 2, there's a drop in PK. And at the same time, the classical ADA assay shows you a signal. And again, the titer is not that high. And what's interesting is that actually it goes down over time. It's still confirmed positive, but the titer is at the edge of detectability. So then we ask the question, what about IgM? And this time, we clearly see IgM. So it's distinct from this other subject. And we also did subject now this patient to the specificity analysis, and it also looks different now. So again, this is your starting point. With COVA322, we get it down to 0. Now with the mixture of adalimumab and the specific FynomAb, there's only a minor contribution. And the same minor contribution you can also see just by the specific FynomAb. Adalimumab on its own, this time, is not having an influence. So here my take-home message is that here the response is not adalimumab, but it's also not just the specific FynomAb. It seems to be a combination of a response that is directed against the FynomAb and the linker. The linker also plays a role if you look at the two controls. I don't want to go too much into detail, but the take-home message is the picture is different. So we do see an early response. Here we do see IgM, it's transient, and the specificity is different. So when we did see such findings that we early lose basically the exposure, we asked the question, so is the molecule and is the bioactivity truly gone? Or could it be that we have antibodies that interfere with our PK assays because we have sophisticated PK assays where binding to the targets could have an influence and that there's maybe still some bioactive molecule around. So we did use again our bioactivity assay, and this is just to show that this nicely works in a dose-dependent manner, and you could actually use this assay also to quantify remaining concentrations of our candidate. And we did again look for two extremes. So this is one where it nicely looks the PK and you lose your exposure later on. And we did look in our bioactivity assay at week 2, 4, 6, 8, 10, how does the bioactivity look like? And it's quite clear. So we have full bioactivity until week 6, and by week 8 we completely lose bioactivity, and that basically matches your exposure. And the same is true for this other type of response. So here we are losing exposure quite early. So again, your exposure and your bioactivity matches quite well. What we just, as additional information, what we also addressed is the question whether the anti-drug antibodies are neutralizing or non-neutralizing. And I just can tell you that for this IgM response, actually they seem to be non-neutralizing. Here they are neutralizing, but at the end it doesn't matter because both are clearing. Both are inducing removal of your compound from the circulation. So at the end it doesn't matter. Okay, and this actually brings me to the end of this presentation. Just some high-level summary. So when you do your assay planning, then I would advise, if you have a complex molecule, that you start with assays that allow you to make the discrimination, is the molecule not just present, but is this also fully active and is it intact? But that requires sophisticated assays and that requires some front loading of work that you should invest. And the same is true for immunogenicity. So in addition to just doing your classical anti-drug antibodies, it helps you if you can learn against which parts are your anti-drug antibodies directed, especially when you are dealing with a novel format, because it could be, so luckily for us, in none of the subjects, we had ADAs either pre-existing or ADAs against our scaffold. So if this would be the case and you have the data, then you have to think hard, how can you re-engineer your technology to get it out? So here it looks like that we simply, the molecule on its own, in some cases, gets a signal. But to allow you to make such conclusions, you need a particular assay and to generate data. And the last bullet point, that's even if you have exploratory assays that are not fully validated, don't run under GLP, GCP, that still can help you quite a lot, especially in early development, in making good data-driven decisions. Most important slide, acknowledgement. So basically the half or more than half of the entire Covagen folks have heavily been involved in this project, and we also work with external consultants. And here in particular I want to mention Jennifer Sims, who helped us with the non-clinical. And now, final slide. Now it's time to ask for hard questions.